it's my great pleasure to introduce our second speaker for today, Alessandra Salerno. Alessandra is also in the same room in Dundee, in the University of Dundee. So this is great to have you both today. And Alessandra has been a postdoctoral researcher at Center of Protein Degradation at the University of Dundee since 2023 under supervision of Alessio Ciulli. She earned her PhD in medical chemistry in the University of Bologna in Italy in 2022. And currently she's funded by Marie Skłodowska-Curie Fellowship. Her wider scientific interest spans over the proximity labeling methods and probing protein-protein interactions, as well as discovery of anti-infective agents. Thank you very much for joining us, and we are very much looking forward to your seminar.
Thank you, Miko. Thank you, everyone. And it's a great pleasure to be here to this seminar. And I'm really excited to show with you and to share with you this our latest work on FerroTAC, harnessing Ferrocene to the design of dynamic PROTAC linkers. So I do believe that I don't have to explain to this audience what PROTAC is, especially after Charlotte's great talk. But I just want to point out here and highlight, as we know, that target protein and E3 ligase ligands are chemically linked by a linker. However, from a medicinal chemistry perspective, the linker does not always connect to two parts of the molecule, but it has a role in stabilizing ternary complex and productive ternary complex formation, but also influencing PROTAC potency, selectivity, and ultimately also modulated pharmacokinetic properties. To give you some numbers, so this is the update data from PROTAC DB, a database on PROTACs, and we see that on average there is a new linker introduced every 2.2 PROTACs. This means there is a huge ongoing effort in understanding and optimizing PROTAC linkers to unlock the degrader's full therapeutic potential. So to give you some context, PROTAC were initially historically designed by incorporating linear aliphatic or PEG linkers, as we find in DBET6 or MZ1. Then we have shifted towards more rigid and heterocyclic scaffolds in order to modulate the pharmacokinetics, specifically the solubility or the stability of the compounds. We also face the incorporation of more complex chemical architectures, such as the microcycles or trivalent branch linkers. And from last year, I was involved in this terrific work led by Claudia as postdoctoral research here in Alessio's group, where we leveraged the reversible linkers based on thiol disulfide exchange chemistry in order to streamline the identification of active degraders by dynamic combinatorial chemistry. So what I'm trying to say here, I think, is that linkerology is not only a way to fine-tune degrader activities and properties, it's also a way to boost medicinal chemistry exploration in order to address what are the PROTAC limitations, such as mainly the poor permeability, the susceptibility to the efflux, or their identifications. So in this direction, we and others have already kind of addressed these issues by exploring different linker design strategies, such as the replacement of amides to esters, but also by involving new linker compositions and exploring new linker composition that could influence the conformational dynamics of PROTACs, namely boosting their chameleonicity. So the chameleonicity can be explained as a feature that the molecule has to change their structure in response to environmental conditions. This feature is known since the 70s, but the interest in this concept has grown significantly in the past decades because we do see a drug discovery shift towards novel chemical modalities such as PROTACs and others. The chameleonicity is a feature that, in fact, is been shown to be crucial also for PROTACs, since they can adopt more folded conformation in apolar environments such as the cellular membrane, and this can allow to achieve a higher cellular permeability. So we do know that linker design can enable PROTACs to act as a molecular chameleons, relying and building on non-covalent interaction, such as pi-pi stacking or more commonly intramolecular hydrogen bonds. So in this work, we wanted to explore unconventional linker moieties such as the Ferrocene, and we identified the Ferrocene as a promising unconventional moiety for the development of FerroTACs as a novel strategy for bias and boost PROTAC chameleonicity. So for those of you that are not familiar, as I was when I started this project with Ferrocene, Ferrocene is a sandwich-like complex. It's an organometallic complex that has an iron that is tightly bound in between two cyclopentadiene ring. It was discovered in 1951, and then the structural confirmation came four years later. And the Nobel Prize in Chemistry was awarded for the discovery of Ferrocene in 1973. So the compound has a very high stability and a tunable toxicity thanks to excellent redox properties. As a structure is being used and versatile, it's proved to be absolutely versatile in different platforms and in different fields, including and not limited to medicinal chemistry. In fact, even if it seems a bit exotic and very niche, the Ferrocene structure is known to the medicinal chemistry community and has been successfully incorporated in different bioactive compounds, such as the chloroquine, and that is then with the incorporation of the ferroquine takes the name of ferroquine. And this is now reaching phase 2 of clinical trial as an antiplasmodial drug. So the Ferrocene has been proved to be highly stable in biological environments, and it's generally incorporated to improve the lipophilicity and to enhance the cytotoxicity when needed. However, what we were more interesting about the Ferrocene is this Ferrocene ball bearing feature, which means that the Ferrocene is the cyclopentadiene ring are freely rotating in solution because the energy barrier in between the cis conformation and the trans conformation is very low. So we do believe that, especially when the Ferrocene bears a substituent, this could favor the cis conformation thanks to weak interactions. And those weak interactions can be controlled and by external factors such as water, and they can shift the preferring conformation into a trans conformation. So our question was, can we leverage Ferrocene scaffold to enable PROTAC dynamic conformational changes? And inspired by those properties, we try and incorporated Ferrocene as linker moiety in bifunctional compounds, obtaining those FerroTACs that we wanted to exemplify this concept with FerroTACs. And we produced PROTACs bearing Ferrocene that addressed 3 different systems. So starting from the design and synthesis, as I said, we wanted to leverage VHL homosystem, VHL against Celebron, and based our design on the classic MZ1. So we did incorporate the Ferrocene as linker moiety, obtaining a small library of FerroTACs. For the from the chemical point of view, we use highly accessible and affordable Ferrocene building blocks that are allowed either symmetric than asymmetric functionalizations. So once we had the compound in hand, we wanted to check by NMR studies. We wanted to look into the conformation of those compounds. So to do so, we search for weak intramolecular hydrogen bond patterns, looking at amide protons in the molecule. The first assay that we run is based on the solvent accessibility. We basically recorded the spectra of the compound in two different solvents. The first one is an apolar chloroform solvent that mimics the cellular membrane. And then we have a more polar DMSO solvents that resembles the extra and the intracellular space of the cells. So what happens is that by switching from one solvent to the other, by adding DMSO, this can disrupt the weak intramolecular hydrogen bond pattern, and this results in the chemical shift differences in between the two solvents. This chemical shift is then converted in these values, also known as ANMR, that can give us a quantitative assessment of the intramolecular hydrogen bond. So as a result, if we are looking at strong intramolecular hydrogen bond, those are kind of locked in the structure, so we won't have any change in terms of chemical shift, while weak intramolecular hydrogen bond show a larger differences between solvents. So here we have the two spectra. In red we have the chloroform, and then in blue the DMSO. So what we found from here, what we noticed at the very beginning, is that the amide number 2 doesn't change in between the two solvents. So it's likely locked in a strong intramolecular hydrogen bond. And this is aligned with what was already reported in 2020 by crystallographic data. By looking at the amide next to the ferrocene, then we kind of we found that the NMR scores were in line with the presence of a weak intramolecular hydrogen bond. This means that there is a network here of intramolecular bonds that are likely mediated by the ferrocene. As a second assay, we also wanted to gain an extra level of information, so we performed variable temperature NMR in chloroform. So in this case, we are looking only at the apolar conditions. So we are only in chloroform. By raising the temperature, what happens is that weak hydrogen bonds are more likely to break, resulting in shift of the chemical signals. So again, if we have strong intramolecular hydrogen bond, those won't shift while weak will. So here we have the spectra of the compound recorded at 10 different, increasing the temperature of 10 degrees each time. And what we see again is that the number 2 is definitely involved in a strong intramolecular hydrogen bond, while the amide 3 and 4 are support the hypothesis of those to be initially shielded in an apolar environment, and this is favored by a cis conformation of the ferrocene. So just to give you like a graphic idea of what I'm talking, we do believe that the amide 3 and 4, according to our speculation, are involved in intramolecular hydrogen bonds where 3 can create a bond with the carbonyl, while the amide number 4 it's kind of more stabilized towards 2 different acceptors of the hydrogen bonds. So the conformation analysis overall by NMR supported the idea that FerroTACs can adopt a more compact conformation in non-polar solvents such as chloroforms and acted as molecular chameleons. Of course, by addition of water as a strong hydrogen bond acceptor, we are likely able to shift those equilibrium towards a more linear configuration. So once we had the bias of the conformational bias of those compounds studied by NMR, we wanted to check if those molecules will still retain in PROTAC mechanism of actions. So we did check for degradation assay by Western blot. And for the system of like HOMO VHL, we can say that we could see that our compounds bearing the ferrocene had a dose-dependent and a time-dependent degradation, which were very comparable to the reference CM11. The same happened with the hetero system, so Celebron VHL system. We can see that the ferrocene compound AS4 induces the degradation of Cellular as good as for the reference that we had. And same for the MZ1 analogs. So the DC50 and the Dmax, as well as the kinetics of the degradation, was comparable to that of MZ1. So all the FerroTACs showed degradation potency, rapid onset, and also ubiquitin and proteasomal dependent degradation, which I'm not showing you today. So as part of the mechanism of action, we also wanted to check for ternary complex formation. And to do so, we leveraged native gel electrophoresis assay, as we previously reported. So the assay worked by incubating the recombinant protein with the PROTAC and then loaded into a non-denaturating gel. So what we see here is that the ternary complex formation is monitored by the upshift of the bands that is aligned with the increasing size of the complex. So basically, in the lower band, this is corresponding to VCB complex alone or in a binary fashion, while where we add either CM11 or our two FerroTACs, we have a complete transformation into the ternary complex. As I mentioned, the Ferrocene compounds can somehow have some cytotoxicity, so we wanted to rule out the cytotoxicity as part of the mechanism of action of those compounds. And to do so, we checked cytotoxicity assay in 2 different cell lines, leveraging DQ1-based degraders. So as you can see here in HT116, there are BET-sensitive cell lines. We do have some cytotoxicity up to 10 micromolar. However, the cytotoxicity isn't from the FerroTAC AS6 is not as much different from the one from MZ1. So we kind of believe that the degradation we see in HT116 is more linked to the degradation of the target rather than the cytotoxicity itself. In fact, when we switch into Action 293, there was no degradation. There is degradation, but there is no cytotoxicity. In fact, the FerroTAC cell viability is similar to that of MZ1 up to high concentrations. So with the mechanism of action evaluated in terms of PROTAC and degradation and ternary complex formation, we wanted to experimentally evaluate the permeability of those compounds. So we do know that sometimes in vitro technique can have some drawback, especially from PROTAC. So we preferred to evaluate the cellular intracellular availability by nanobreath target engagement assay. So in this case, the assay works in 2 different setups. So we have in the live mode, the membrane can impede the compound to access the target. So the target is the compound that we want to check the permeability of. It's kind of limited by its own permeability. However, the same experiment is then run in permeabilized mode, where the barrier by the cellular membrane is removed. So we are checking here for intrinsic affinity of the compound. Ideally, if the compound is highly permeable, the curve in its permeabilized and live mode is overlapping. If there is a right shift in between the live and the permeabilized, it means there is a lower intracellular availability. So, for example, here I'll show you the data on MZ1. We do see that MZ1 in the live mode has a right shift curve compared to the permeabilized. So this suggests that MZ1 has a very poor cellular permeability and is limited by intracellular availability. However, when we did the same for our compound AS6, the differences in between the live and the permeabilized mode is much smaller. So here I'm showing the data only on this system, but overall, all the FerroTACs demonstrated smaller differences in VHL engagement between the live and the permeabilized mode, suggesting for those compounds to have a higher intracellular availability, which is linked to unimproved cellular permeability. Another aspect that we wanted to check was the impact of efflux transporter or those compounds. So it is known that some PGP efflux pump can limit PROTAC efficacy, and also they represent drug resistant mechanisms. It is also well established that the count in the compound hydrogen bond donors and consequently the polar surface areas are some of the elements that determine the susceptibility to PGP efflux. So our idea here is that by pretreating the cells with the PGP inhibitors, like zosukidar, if the compound is normally subject to the efflux pump, it would after pretreatment with this inhibitor, we would have an intracellular, an improved and increased intracellular concentration, then is then mirrored to an improved degradation potency. So by checking a degradation, this is a hybrid degradation assay with or without pretreatment with zosukidar. What we see is that MZ1 gains in DC50 almost 6 times, as an improved DC50 of almost 6 times when pretreated with zosukidar. This let us think that MZ1 is normally subject to PGP efflux. However, when we checked for the FerroTACs analog AS6, the differences in between the DC50 with or without pretreatment with the inhibitor is less, it's much smaller. So we the take-home message here is that compact conformation that can be allowed by the Ferrocene dynamic movement can not only improve permeability by passive membrane, but can also reduce the susceptibility of those compounds to PGP efflux mechanisms. And as last piece of information that we wanted to check and calculate some in vitro parameters in terms of physicochemical properties. So we checked at first for lipophilicity, and we measured either the log D values chromatographically or experimentally. And what we found is that the incorporation of the ferrocene does increase and enhance the overall lipophilicity in all the system. But this is not surprising because this is aligned with what has been reported in the use of ferrocene in the medicinal chemistry. So when we checked for solubility, we could see that overall the solubility of the FerroTACs is reduced in PBS. However, it demonstrated that there is improved solubility at pH 5.8 in 2 out of the 3 systems. And as a last parameter, we also checked for plasma and microsomal stability. And we could see that here the trend is it's a bit general, so it's really system dependent. Like in the VHL homoprotac, the FerroTACs was not, the FerroTAC mouse plasma stability was not affected, but while in the back domains, it has an half-life that is much shorter than one of them Z1. However, for microsomal stability, all the FerroTACs displayed 2 to 10 greater microsomal stability in compared to the linear analogs. So to conclude, I kind of believe that we have demonstrated that Ferrocene is a versatile strategy for designing PROTAC linkers, that they can foster intramolecular interaction that results in more folded state, and is allowed to improve the cellular permeability. On top of the cellular permeability, also it can reduce the efflux liabilities, and it retains the robust target degradation of the linear references as well as their favorable pharmacokinetic properties. As a general, as I said, we use the ferrocene and we kind of prove our idea incorporating into products. But as a general idea, ferrocene dynamic behavior could be used to leverage any kind of protein-protein inducer, such as molecular glues, or any other kind of induced proximity mechanisms that are not limited to degradation. So just as a take-home message, sometimes think outside the box can actually lead to less opportunities and new big crazy ideas. And to conclude, so I want to thank everyone that has worked with me on this project, Claudia and Liane specifically, and of course Alessio as a great supervisor and a bit of a crazy ideas generator, and everyone involved in the center and working at the Center for Targeted Protein Degradation and my funding agency. And I'm happy to take any questions.